Okay, so this is question 2. Define, so this is just recall, so competitive inhibitor, after 2 marks for each, is a similar shape to the substrate. It can therefore bind to the active site of the enzyme, and when the inhibitor is bound to the active site, then it's going to prevent the substrate from binding. If we have a non-competitive inhibitor, then that binds to a region of the enzyme other than the active site, which is called an allosteric site. And by binding to the allosteric site, it changes the shape of the enzyme and therefore changes the shape of the active site so that the substrate will no longer fit. So they both inhibit enzymes because they reduce their activity. Between A and B and C and D. So between A and B, we're obviously increasing the temperature, and we can see the rate of reaction is increasing. It's not asked for a description, so we're not going to get any marks for that, so we've got to explain it. So as the temperature increases from 10 to 40 degrees centigrade, the kinetic energy of the particles in the solution will increase, and therefore there will be more successful collisions between enzyme and substrate, and more enzyme-substrate complexes formed. If we look between C and D, we've got a rapid decrease in the rate of reaction. This is due to the high temperatures affecting the three-dimensional shape of the molecule. Basically the hydrogen bonds, etc., that are holding it together are going to break, so we're going to lose its complex three-dimensional shape. The substrate will no longer fit into the active site, and therefore the rate of reaction decreases, and once the enzyme is denatured, we can have no rate of reaction because the substrate will not fit.